Hello students, this is Inheritance and Variation Part 2. I have prepared one video and uploaded it that was Inheritance and Variation Part 1. In that part, we have introduced, I gave brief introduction of whole lesson, what are the important points and the new terms which came in this lesson, that is what is inheritance, what is variation, what is heredity, the genetics branch of science again, and Mendel's experiment, why he was successful, the again the seven traits which Mendel studied. Okay, so all that part we have studied in the part 1. It was not that much brief introduction of the lesson, but it was like what are the important concepts you are going to study in the whole lesson that I was trying to tell you in that part. So in this part 2, inheritance and variation part 2, I'm going to introduce you Mendel in a very brief way, because it's very important to you to understand what was his work, why he selected pea plant, why it was necessary to study the plant. Okay, why he gave the laws, what is the importance of his experiment. So all the things you will come to know, but before going to that, we will have a brief introduction of the scientist who gave the important laws, that is basic laws of inheritance. So Mendel was a scientist, and he was born on 22nd July 1822, and he took his higher studies in physics, mathematics and natural sciences. In 1856 he began his famous hybridization experiments on the garden pea plant. That pea plant you have studied, I have shown the slide what how the garden pea plant it looks like, and what are the seven traits that he studied, and the hybridization experiments what he did with, so with that plant. So in 1865 he presented his work in a paper entitled Experiments in Plant Hybridization. Unfortunately his work remained unrecognized. So why it remained unrecognized, and what are the drawbacks, what are the reasons, more particularly what are the reasons behind that we are going to study in this part. Later his work was noticed by few scientists again, and they proposed the chromosomal theory of inheritance by using the Mendel's data on his experiments. So why his work remained unrecognized? So there are a few points which I want to clear with you, I want to discuss with you in detail. So his work was not widely recognized. His approach of using mathematics and statistics to explain the biological phenomena was totally new, and it was unacceptable to the biologists at that time. So we have studied in the part 1 that Mendel's success. He successfully experimented with the plant, and he recorded the data when he hybridized the traits, hybridized the plants, okay, pea plants, and he collected his data, he recorded the data in mathematical and statistical way. So because of that there was too much accuracy in his experiments, but this concept was totally new to the biologists at that time, and so they never accepted the Mendel's experiments. Another part was the variations were observed in nature, and Mendel's concept of factors, that is genes now we call it, as a stable and discrete unit which controls the expression of characters, and that a pair of alleles did not blend with each other, was not accepted by his colleagues, or at the time of scientists. Okay, so what Mendel introduced, that the genes are something like factors, he called that factors. The factors, they are getting transferred from one generation to the next generation, and because of the transfer of the flow of the factors, the inheritance or heredity is going on. Okay, so it is carried out by the discrete unit or stable unit that he told, or he termed as factors. So that concept was totally new to the biologists at that time, and so the biologists, they completely avoided Mendel's experiments or his research, and he also did not know the physical locations of the factors in the genetic cell. So that concept was totally new at the time. Mendel was unknown about what is getting transferred, as microscopes were not available at the time. DNA was not invented at the time, so he was not aware about the chromosomal part or the gene which was getting transferred. So that part, because of these all criteria, or because of all these reasons, all the biologists or scientists, they avoided Mendel's experiments. So after that, the chromosomal theory of inheritance, it came, and few scientists, they discovered the Mendel's work. So the scientists Hugo de Vares, Correns, and Vaughan, they independently rediscovered Mendel's work on the inheritance of traits, and it was possible because of the advancements in the microscopy, which was done at their time, and scientists were able to observe the cell division and the structure of chromosomes under the microscope at that time. So the after discovery of microscope and structure of DNA and genes, the Mendel's work was rediscovered by few scientists, and based on the observations, based on his data, the scientists, they put forth the chromosomal theory of inheritance, and it was put forth by Sutton and Boveri. This theory identifies the chromosomes as the carriers of genetic material. So this is why the chromosomal theory of inheritance is supposed to be the basic fundamental principle in the biology. Okay, so in the this, what is explained exactly in the chromosomal theory of inheritance. Now we are going to study the chromosomal theory of inheritance in the next videos, but let's have a short or quick look on what is chromosomal theory of inheritance, what why it is important to study, or why it is called as the fundamental principle which was proposed by this chromosomal theory of inheritance. So this theory, it states that the chromosomes which are present in pairs in somatic cells. Now somatic cells, we have studied in the cell division in the 11th standard part, that somatic cells, these are nothing but the body cells. So every living organism contains the somatic cells, and each somatic cell, it contains chromosomes. During the gamete formation, homologous chromosomes pair, segregate, and they separate independently during meiosis. So we have studied this part too. I'm going to explain this part in much detail, that how the chromosomes they play a very important role during human reproduction and all. And not only this part, but also in case of in case of pea plant, that is Pisum sativum plant, how the segregation part or gamete formation part, or the somatic cell part, homologous chromosomes and independent assortment and all, we are going to study in detail. So let's move towards the next slide. So moving to the next, we are having few questions on this slide, that you'll come to know these answers of these questions, okay, when you are going to study this part. I'm having few diagrams here if you're observing on the screen. Okay, so do you know what are the sex chromosomes? Where the gene is situated on the chromosome? What decides the characters of a child? What happens to the chromosome during reproduction process? And where exactly heredity plays its role during the reproduction process. So we are going to study all this part in detail. But before going to that part, if you remember the part 1 of inheritance and variation, I have explained a few characteristics of plant. Let's take an example of a plant, that flower, color of flower. I gave an example of white flower and violet flower, or red flower and white flower, how the characters they get passed. Or we took the example of expression of characters like color of hair, curly hair, color of eye, complexion, that parents characters of the parents, they get expressed in their offspring. So that all we have studied. So why it is possible, means how it's possible. Okay, so what are the sex chromosomes? Who decides that which chromosome, which part of the chromosome decides that which character or which trait will get expressed in the offspring in F1 generation? Who decides that which is the dominant character and which is the recessive character? That we are going to study in detail in this slide. There are two images. Okay, this is first image where the set of chromosomes are shown. Now this is first set of chromosome 1, 2, 3, 4, 5. As you can see the numbers till 22, all the chromosomes are in pairs, and this is the last chromosome where XX chromosome is there. X is both the chromosomes, these are XX chromosomes. Okay, so out of these, how many chromosomes you can see on chromosomal pairs you can observe in this image? There are near about 23 pairs of chromosomes. So out of these 23 pairs of chromosomes, 22 pairs are autosomes, while the 23rd pair, now this is 23rd pair, which is a sex chromosome. This sex chromosome or pair of chromosome, it decides the sex of the offspring or sex of the zygote. Now apart from this last pair of sex chromosome, till chromosome pair 1 to 22, all these chromosomes, these are called as autosomes. What are autosomes? Autosomes, these are the set of or pairs of chromosomes which do not determine the sex. The sex chromosomes are those chromosomes which determine the sex of the individual. Now, what is diploid cell and what is haploid cell? Now, you are going to come across this. You know it. You have studied it your previous standard. In your 10th standard, you have studied it. In 11th standard, you have studied it in the cell division part. So, diploid cell is having 2n number of chromosome. Now this is the diploid cell. You can see the diagram. It contains 2n number of chromosome. N, it stands for number of chromosomes. Okay, and haploid cell contains the n number of chromosomes. N number of chromosomes, that information which is present. So in the haploid cell, the double chromosomes, they are not present. Now you can see, if you are observing the first diagram or first chromosome, which is very large, or length is it is completely visible. Now this is 1 and 2. So this is pairing of two chromosomes. When you talk about a sex chromosome, or when you talk about haploid chromosome, when you talk about haploid or n number of chromosome, in that n number of chromosome, one chromosome will be there. Okay, so there will be absence of two or paired chromosomes. That is the difference between the diploid cell and haploid cell. Hope you understood autosomes. What are autosomes? What are sex chromosomes? Autosomes are not they don't decide the sex of the offspring, while sex chromosomes, this is the 23rd or last pair of the set of chromosomes which decides the sex of the offspring. So let's move toward the next slide. Now, position of gene, where the gene is situated, as the Mendel suggested that something was getting transferred in his experiment when he studied Pisum sativum plant. So something was getting transferred from one plant to another plant. That was his conclusion. So if you observe the first diagram, this is eukaryotic nucleus, in a eukaryotic cell. In the eukaryotic nucleus, when we zoom in, now this is the zoom in part where the cross sections, you can see these threads, these are nothing but the chromosomes. Okay, so this is the chromosome. Now this is a nucleus cell. In the cell there is presence of nucleus, and this nucleus contains this chromosome, which contains X type shape of structure. When we unwind this DNA, as the DNA is or the chromosome is made up of tightly packed DNA. When we unwind the chromosome, the DNA is a thread-like structure. Now this is a DNA which is compactly arranged inside the chromosome, and these chromosomes, numbers of chromosomes are again compactly arranged inside this nucleus of a cell. Okay, so now where the gene is situated, see when you are able to see that thread of DNA. Now you will be able to understand. Now this is the segment. Okay, now this segment, this part is called as a gene. When we cut the DNA or the genes, where they are situated on the segments of the DNA. They are partly arranged on the DNA. These are the segments of the DNA. You'll understand it better. For example, now this is one encyclopedia. This is one book. Okay, this is 2, 3. As you can see the numbers. Now each book is nothing but, just imagine that each book is a single gene. Now this is a first gene, this is second gene, this is third gene. Okay, so likewise all the genes, they are arranged in this manner on the DNA. Now this complete encyclopedial chain, it forms a complete DNA. Now you can see you can observe the sex. Now this is male and female. Okay, so this is male and female chromosomes which come together to form a zygote. We are going to study it, so I hope you understood position of gene. Gene is situated on the chromosomal part. So let's move toward the next slide. So in the current slide, we are going to understand that how the sexual reproduction occurs in the human body. Now this diagram, there are a few diagrams over the screen. Okay, so in this diagram, you can observe that there are 23 pairs of chromosomes which are present inside the human body. Now, there are two flow charts you can observe. This is the maternal side this is the paternal side where the father, it contributes a sperm, which is haploid in number, and the zygote get formed. While in case of mother, ovum get formed, and by the fusion of sperm and ovum, there is formation of zygote. So how it occurs? So normally, the human body, it contains 23 pairs of chromosomes. So out of these 23 pairs, 22 are autosomes and 1 is sex chromosomes. So observe this diagram now. So in this image, you can observe that there are paired chromosomes from 1 to 22. All these 1 to 22 chromosomes, these are known as autosomes, while this last pair, it is representing X and Y. So this it contributes in sex determination. So this is known as sex chromosome, pair of sex chromosome. So paternal side of father, he gives sperm which is haploid in number. So the diploid, each and every cell is diploid, but in case of gametes, when they form gametes are always haploid. So father, the sperm is a male gamete which is haploid in number. The ovum is a female gamete which is haploid in number. So this haploid cell, it contains 22 autosomes and 1 sex chromosome. That is, it contains 23 chromosomes. While in case of ovum, it contains 23 chromosomes. Out of these 23 chromosomes, these 22 are autosomes and 1 is sex chromosomes. So when there is fusion of sperm to ovum, when there is fertilization, there is formation of a zygote which is diploid in number. It contains 2n number of chromosome, and now this 2n number of chromosome, it is each and every cell is having 44 autosomes and 2 sex chromosomes. So at the end, the zygote, it contains 46 chromosomes. Okay, so hope you understood that from a diploid cell, haploid gametes are formed, and these haploid gametes, when they get fused, they form a diploid zygote. So you can observe here haploid cells, these are always having n number of chromosomes. See, these are the n number of chromosomes, they are single. And in diploid cells, the chromosomes, they are in pair. Okay, so these are present in pairs in case of diploid cells. So when you observe the third or another diagram, this is having homologous chromosome. In case of homologous chromosomes, if you are observing the first chromosome, it is from the dad side, it is paternal, it is carrying the characters of the father. Now second chromosome is from the maternal side, from the mom, it is carrying all the characters which are coming from the mom side. So these lines, if you are observing, these lines are present at the same position. The positions are same. Again, these genes, now these lines, these are called as position of the genes. Okay, loci, locus. And now these paternal side, this gene is carrying information of eye color, and again this chromosome which is from the maternal side, it is also carrying the information from the mother side. It is also representing the eye color. Now talking about the hair color, it is from the paternal side, again the same position of the genes, and this hair color, it is coming from the maternal side. Okay, so homologous chromosomes means what? Homologous chromosomes, these are similar. Homo means same. What is the similarity? These are morphologically similar, they are structurally similar, and they are genetically similar. So morphologically, structurally, and genetically, when the chromosomes are same, genetically means if they are having the same genes which are positioned at the same positions, and these genes are carrying the same information, means this position, this gene is carrying the information of eye color, so this gene will also carry the information of eye color. Again, this gene, if it is carrying the information for hair color, then this gene will also carry the information of hair color. So that if this characteristics are same, then such type of chromosomes, they are known as homologous chromosome. So moving to the next slide. Now what are alleles? What are alleles or what are allelomorphs? So these are the variety of genes which are present on the chromosomes. These are called as alleles. Now coming back to our example, in the previous slide we were talk about the characters which are present in the human beings, but as in this Mendel's lesson we are talking about the flowering plant, that is Pisum sativum. So for example, this chromosome, it comes from the plant which forms purple flowers. So this gene, it is carrying the information or allele for the purple flower. Okay, and this chromosome is coming from another plant, that allele for the white flower. So this chromosome is having the information which expresses itself in the form of white flowers. Okay, so if you are observing, the size of the chromosome is same. Okay, the position, these genes, they are positions, the bands, if you are watching the colorful bands, so their position is same. Okay, so locus of the color flower color flower of the color gene. Okay, so this information is again same. So these two are morphologically, structurally, and genetically same. So these are known as alleles. Okay, homologous chromosomes. So let's move to the next slide. So going to the structure of chromosome, you can get a question on this. Structure of chromosome, will have a close look on that. So in this slide, you're having these two images. First is the structure of chromosome. Here, part A is one parts of the chromosomes are shown in this diagram. While in the second diagram, it is showing the secondary constructions, which are or this chromosome in detail, how it is made up of. It is the structure of chromosome. The question can be asked from this part separately. And the structure of X and Y chromosome in humans, the structure of X and Y can be asked. So we are going to study this part in detail. Okay, and I will explain what is it, what are these diagrams are. Okay, so before going to this structure, what is the meaning of chromosome? Chromosome, it contains two parts. Okay, first is chroma and next is zoma. Chroma means color and zoma means body. So these chromosomes, these are filamentous bodies present in the nucleus. We have studied, we have observed in previous slides that how the DNA is compactly arranged inside the nucleus. Okay, so it is very filamentous and it is present inside the nucleus. It is visible only during cell division. So chromosomes, they are having the ability to self-replicate, and they play important role in the heredity, variation, mutation, and evolution of the species. So the chromosomes which are morphologically and genetically similar, they are called as homologous chromosomes. Homologous chromosomes we have studied in the previous slide also. We have discussed it, that the homologous chromosomes, they are morphologically as well as genetically they are similar. Okay, apart from that, size of the chromosome. what is the size of the chromosome. So the size of chromosome, it varies from species to species. In metaphase, each metaphase, the chromosome, it varies from 0.1 to 33 micrometer in length and 0.2 to 2 micrometer in thickness. So the length and the size of the chromosome, it varies. Now, what is the shape of the chromosome? The shape also varies according to the stages of the cell division. So when we have studied the part of cell division, in that part we have studied that the chromosomes, they are having different kinds of structures. Okay, so they're according to the stage of the cell division, the structure or the shape of the chromosome, it get changed or it varies. So during the interphase, the chromatin network, in metaphase, it shows, the chromosome shows maximum condensation and it appears very short and very thick. So structure of chromosome. Now this is the structure. Now metaphasic chromosome, it has two identical halves. Now these are the sister chromatids, which you can see. Okay, now these are the sister chromatids. If you're observing it, these are the sister chromatids. Now, each chromatid is a turn made up of subchromatids. These are chromonemata. Now, this is chromonemata, which is present inside the chromosome. And these chromatids, they are attached at the central point, at a certain point, and that part is, one point is called as centromer. Okay, so where they are attached, that part is called as centromer. Now this is a centromer where these two sister chromatids, they are attached. Now this is a matrix. Okay, so coming to the next in detail structure of the chromosome, you can observe that these are the parts of the chromosome. Now this is a satellite and nuclear organizer, then satellite constriction, secondary constriction, nucleolus, primary constriction, secondary constriction, chromomers and telomer. If the question is asked on the structure of chromosome in detail, you have to draw both the diagrams. If the diagram are asked separately and you have to describe it, you have to write two points each. You have to describe each and every title of the or labeling of the chromosome. So the centromer is also called as primary constriction. If you're observing, this is a centromer and this is a primary constriction. So the centromer and the primary constriction, they are one and the same. Okay, so the centromer is also called as primary constriction, and during cell division, the spindle fibers, they are attached at the centromer. At this position, they get attached, say, as spindle fibers. Secondary constrictions, they are also found to be present on the chromosome, and these secondary constrictions, these are the nucleolar. They are also called as nuclear organizers, and these are the nucleolar organizers, and they are also called as secondary constrictions. And these are known as nucleolar organizers because they are helpful in the formation of uh, because they form the nucleolus. Now, after this nucleolar structure, as it forms the nucleolus, the part of the chromosome beyond the nucleolar organizer is very short, spherical, and it is thick, and that part is called as satellite. The tip of the chromosome is called as telomer. It is the tip of the chromosome. The surface of the chromosome bears large number of swellings, and these swellings you can see inside the chromomers. Now these are the chromomers inside the chromosome. These are the swellings which are present. Now what is the function of this uh, chromosome, chromosomal part which is having telomer? So this is the telomere. What is property of the telomere? The telomere, it prevents the attachment of the two ends of the chromosome. Okay, it keeps the ends aside. So this is the detailed structure of the chromosome. Now there are types of chromosomes. So this is the position. You can see the diagrams, 1 2 3 4 diagrams are there, and the blue or violet color dot is nothing but the centromer, the position of the centromer. On the basis of that, the chromosome is divided into 4. This is called as metacentric. It is the centromer if it is positioned at the middle of the chromosome. Now this is called as submetacentric. Towards it, submetacentric is the some or little distance away from the middle point of the chromosome, while in the acrocentric position, when the centromer we are talking about, you can observe that it is present just near to the end of the chromosome, while in case of telocentric chromosome, the chromosome is completely present at the tip of the chromosome. So these are the 4 types of chromosomes which are there on the position of the centromer. Now, so coming back to the structure of X and Y chromosomes which are present in the human, you can observe the diagram. This is X chromosome, this is Y chromosome. Now this X chromosome is longer than the Y chromosome, and these are the non-homologous regions, these are the homologous regions, and this is the centromer, position of the centromer. Homologous region, it is morphologically and genetically same. Non-homologous region, you can see you can observe the difference, right? This part is bigger than this part. Okay, so they are not similar, they are dissimilar to each other. So X chromosome is longer than the Y chromosome. X chromosome, it contains large amount of euchromatin and small amount of heterochromatin. Now what is euchromatin? Euchromatin, it contains large amount of DNA or genes. So that large amount of DNA or genes, they are present in the X chromosome. Y chromosome, it contains small amount of euchromatin and large amount of heterochromatin inside it. It also has Y chromosome, it is genetically less active than the Y chromosome, as it contains less gene comparatively X chromosome. So X and Y chromosomes, they show homologous and non-homologous regions in them. So these are the homologous regions which are morphologically same. These are the similar genes, while non-homologous regions, they show dissimilar genes. They have dissimilar genes. So crossing is possible. Crossing over is possible only in the homologous regions, which are morphologically and genetically same. Non-homologous region. Now, non-homologous region of X chromosome contains more gene than the Y chromosome, and the non-homologous region of X chromosome, it contains X-linked genes, and the non-homologous region of the Y chromosome, it contains Y-linked genes. So why these X-linked and Y-linked genes are present, what are their functions, what are linkages, kinds of linkages, that all part we are going to study in the next lectures. Okay, so let's move toward the next part. So in the previous slide, we have studied that the structure of chromosome and the structure of X and Y chromosome. Now this is a new concept which is included in your syllabus. This is known as ploidy. Okay, ploidy means chromosome sets in a nucleus, and this chromosome sets which are present inside the nucleus, they are divided into 2, that is euploidy or aneuploidy. So euploidy is a common concept. Monoploidy, diploidy, diploidy or polyploidy means having n number of chromosome, haploid 2n number, diploid, and 3n, 4n or 5n sets, or more than 5n sets of chromosomes. If they are present, then that condition is known as polyploidy. While in case of aneuploidy, now what this condition is, it is the presence of an abnormal number of chromosome in a cell. For example, a human cell, it contains 46 number of chromosome. So instead of 46 number of chromosome, if the human cell, it contains 47, or if a human cell contains 45 number of chromosomes, means more than 45, 46 or less than 46, if the chromosome numbers there are present, then that condition is known as aneuploidy. So now that this condition is divided into 2, hypoploidy and hyperploidy. Now hypoploidy, you can understand 2n minus 1 and 2n minus 2. Okay, so hypoploidy is the condition, or it is the state in which the cells, they contain one or more fewer chromosomes. Less, one chromosome is less, or two chromosomes they are less. If such type of condition is there, then that is called as hypoploidy. So humans, they normally have 46 chromosomes per cell, but if a cell has 45 or less than that chromosomes, then that condition is known as hypoploidy. Hyperploidy, see, 2n plus 1, 2n plus 2 means the chromosome number is getting added. So it is a state in which a cell contains one or more extra chromosomes. Humans, they normally have 46 chromosomes per cell, but if a human has 47 chromosomes or more chromosomes per cell, then that condition is known as hyperploidy. Now monosomy, it is 2n minus 1 number of chromosomes if they are present. So I think you have understood that ploidy means the chromosomes of the set of chromosomes which are present inside a nucleus. Euploidy is the, it is common or normally we know all the terms. Okay, this concept is new, that is aneuploidy, hypoploidy, hyperploidy. So this condition, it is when the chromosomes they are less than 46, then it is hypo, and hyper means many number. Okay, more than, normally 46 if they are present, then more than 46 if the chromosomes they are observed to be present in the cell, then that condition is known as hyperploidy. So that's all about this. So hope you have understood today's lesson, you have listened it carefully, and we have started the lesson inheritance and variation in the first part 1, that is inheritance and variation part 1. I have gave you general idea about the Mendel's characteristics or laws, what we are going to study in the lesson, the brief introduction of that lesson and all. And in today's lesson I have introduced Mendel to you, that what is his importance and why these his inventions are important to us, why he studied, how he studied before the discovery of gene, that by naming the gene as factor. And today's lesson we have studied that what is the structure of chromosome in detail, what is the structure of chromosome. Okay, chromosome is playing a very vital role when you are studying genetics. Okay, and when you are studying genetics, the chromosomes and genes, they are very important. So I have explained you the positioning of genes, what the gene is, how it is getting expressed. Okay, so that all we have studied in today's lesson. But I request you to listen the video again and again. Please don't miss the lecture, and please do share the video with your friends, because during lockdown period, as we don't have the book, okay, printed book, we are still having just the soft copy PDF format book, so it's very difficult to study with that part. So please do read the PDF, whatever you have with you, read that matter. I always upload my video within 2 days. Okay, so you get 1 or 2 days of time, and I always try to emphasize the important points. I also give some short, techniques, that what you have to remember. Like today, in today's lecture, the Mendel's introductory part, it cannot be asked in the exam, but the structure of chromosome, now in this part, in today's lesson, today's session, the structure of chromosome you have to practice, because it can be asked. And try to understand the concepts. What is homologous chromosome? The concepts are very new, the biological terms are very new to you. You don't have to there is no need to memorize the things. If you listen it again, if you read the PDF book again and again. Hope so you are studying. So keep studying, keep uploading your comments, that how you are feeling, do you like the video or not. And of course, stay safe and stay at home. So thank you.